and welcome. You are listening to a podcast by the Milner Centre for Evolution at the University of Bath. I'm Professor Turi King, your host, and today I'm talking to Tom Williams, Professor of Computational Evolutionary Biology. Tom uses computational methods to study how molecules, genomes, and microbes evolve, and to learn about the evolutionary history of life. Tom has just been part of a big study trying to understand the evolution of eukaryotic cells, a formative event in the history of life on Earth. Tom, before we get into your study, let's back up a bit. What was life like on Earth before eukaryotic cells?
So, from what we know, we think that life started on Earth pretty soon after the planet's formation. So the Earth is about 4.5 billion years old. That's when the moon was created, and you get the Earth-Moon system as it is today. And based on what we can tell, within about 300 million years of that period, you already had simple cells living on Earth, so what we call prokaryotic cells. So today, the bacteria and the archaea have prokaryotic cells. They're relatively small, so maybe 1 micron across. They don't have a huge amount of internal structure, but all of these rather simple cells, at least structurally, are very biochemically diverse, very metabolically diverse. And so we think relatively early in life's evolution on Earth, within the first, let's say, 1 to 2 billion years of life's history, a lot of the different metabolisms and ways of life that single-celled organisms use today would have already existed. So from things like methanogenesis through to, oxygenic photosynthesis like cyanobacteria and plants do today. On the early Earth, all of these metabolisms would have been evolving in a relatively oxygen-free environment.
So what distinguishes these really simple prokaryotic cells from eukaryotic cells? What's the difference between them?
Yeah, when you look at life as it's organized today, that you've got the prokaryotes, as I've already mentioned, and the eukaryotes. And eukaryotic cells are typically much larger than prokaryotic cells, perhaps 10 to 100 times as big in diameter. In particular, they're very structurally complex. So they're full of lots of internal compartments. The eukaryotic cells have got a nucleus, which contains the genes, which are the blueprint for the cell. They've got mitochondria. Those are compartments in the cell that produce energy. And then in photosynthetic eukaryotes, like in plants and algae, you've got compartments like the chloroplast, where photosynthesis happens. And there's lots of other subcellular structures too. So they're much larger, more complex, and perhaps also more structurally dynamic cells. So they're capable of lots of different sorts of movement. And the other aspect of eukaryotic complexity is that all of the most familiar multicellular groups of life that we know, so animals, plants, fungi, seaweeds, slime molds, they're all made up of eukaryotic cells. So it's not just that individual eukaryotic cells are bigger and more complex than prokaryotes, but also these most familiar forms of life are all made up of sort of agglomerations of large numbers of eukaryotic cells.
This is obviously a really big event in evolution because you go from these really simple cells to these much bigger, much more complex cells that kind of make up, as you say, pretty much what we kind of think of as life on Earth these days, the plants and the animals and us, for example. So how do we think that happened, that step from those really simple cells to these more complex cells?
So we don't know all the details, and the study that we're going to talk about is trying to look at one aspect of that. But I think the sort of fundamental idea here, which has been established now for quite a while, is the idea of endosymbiosis. So the endosymbiotic theory, which is the idea that the first eukaryotic cells came together through symbiosis of simpler prokaryotic cells. So one cell coming to live inside another, and in that way you get sort of a larger compartmentalized cell. And in particular, the idea is that there was a host cell, which was drawn from the archaeal domain of life. And remember, we think of the Asgard archaea, so one group of archaea. And its genome has its descendants today in the nucleus of eukaryotic cells. And then on the other side, we think its symbiotic partner was an alpha proteobacterium. So that's one particular type of bacterium, which evolved to become the mitochondrion, this energy-producing compartment of the eukaryotic cell. And that's a really core aspect of the theory. But beyond that, there's a lot of details that are debated.
So actually, let's go back for a second. What's archaea? Because I think people know what bacteria is, but the average listener won't know what archaea is. So what are archaea?
So archaea are another group of prokaryotic cells. So under a light microscope, they're hard to tell apart from bacteria. But actually, if you look at their DNA or their metabolism, they are very distinct from bacteria. And we actually think that the deepest split in the tree of life is between these two major lineages of prokaryotes, the archaea and bacteria. In terms of their cell biology, the way that they make their cell membrane is a little bit different. And they have a number of kind of unique metabolisms which you don't find in bacteria, in particular methanogenesis. So that's a way of making energy from reacting carbon dioxide with hydrogen. There's some other varieties of that as well. And these archaea, they're globally very abundant and they're found in many different environments. When they were first discovered, which is relatively recently, right, so we're talking about the late 1970s, by Carl Woese and his colleagues, the idea was that archaea were restricted to extreme environments. So they might be living in very acidic environments or very hot environments. But actually, in recent decades, particularly as techniques for sequencing genomes directly from the environment have been developed, we've discovered that archaea are actually found in many different environments, including the same sorts of environments we live in. There are some living in our microbiome, for example. In soils, they're very common. So they're abundant, they're globally distributed, and they're an important part of, life's prokaryotic genetic diversity.
So you've got these two simple cells coming together, one sort of as engulfing the other, but instead of it being munched and eaten, it actually sort of just lives inside this other cell. So what are the big questions here? Presumably it's about timing and how that happened and how these kind of more complex structures within eukaryotes arose, I'm guessing?
Yeah, that's right. I mean, there's a lot of different questions that people are investigating in regard to how that symbiosis took place and its role in the evolution of eukaryotic complexity. But I think one of the big questions is how complex a cell was the archaeal host which entered into this symbiosis with the mitochondrion. And there's sort of a continuum of views of which you have two extremes. At one extreme, which is called the mitochondria early view, the two cells which initially interacted to form that symbiosis were both very simple prokaryotic cells, just like we might see among prokaryotes today. And it's actually the coevolution or the sort of symbiotic integration of the two, the way that they were sort of forced to evolve, to live together and to integrate, which catalyzed the development of all of these complex features of eukaryotic cells. At the other end of the continuum, you have scenarios in which this symbiosis between the archaeon and the bacterium was actually a relatively late stage in the evolution of eukaryotic complexity. And the idea there is that the archaeal host cell might have already had some of the kind of cell biological features that we associate with eukaryotes. Things like, for example, the cytoskeleton, maybe even the ability to phagocytose or to sort of eat food. If you think of the way macrophages or amoeba engulf their prey, that sort of way of eating other cells. And there's a sort of intermediate spectrum of hypotheses you could imagine as well, in which some of those cellular features had already evolved and some came after the mitochondrial endosymbiosis. And a lot of the debate is about the order of the events and the causative role that each of those traits played during the evolution of eukaryotes.
And this is exactly what you've been looking at in your paper. So tell me about your paper. What have you been doing?
Yeah, so what we really tried to do in this paper is to work out the order in which some of these key traits of eukaryotes evolved relative to the timing of the mitochondrial acquisition, the timing of this endosymbiosis. And the way that we tried to do that is by linking the origin of each of these traits to certain genomic features, which we could try to date using some molecular clock or phylogenetic methods. So in particular, we tried to date events known as gene duplications during the evolution of eukaryotes. So when you're thinking about how new functions can evolve in life's evolution, or you're thinking about how complexity can evolve, the idea of gene duplication is one of the key mechanisms. So the starting point would be a single gene in an ancestral organism which encodes some particular function. Let's say maybe it's encoding one of the polymers that's involved in making the cytoskeleton. The idea is that if you've just got a single gene that's playing an important role in an organism, it doesn't really have a scope to evolve because it's got to keep playing that important role. However, if the gene gets duplicated so that you now have two copies, and that could happen because of an error during genome replication, for example, You now have a situation where what's possible in sort of selective or evolutionary terms is different, because now one of those two copies could keep playing the ancestral function that needs to be maintained, while the other could evolve a new function which changes how that cytoskeletal filament forms, or maybe it changes the compartment in which a gene should be expressed. And so as you look at the origin of eukaryotes, we can see by doing comparative genomic analyses that there are a lot of gene duplications in functions that are associated with these eukaryote-specific traits. So what we tried to do in this paper then is identify gene duplication events which we could link to the origin of these traits, and then use molecular clock analyses to try to date them. And then with those dates, we've got the sort of timeline for when each of the features evolved.
So talk me through the molecular clock analysis. How does that work?
The molecular clock hypothesis, the idea of a molecular clock, is that the rate at which genomes change in evolutionary time is constant. So genomes might pick up changes like the ticking of a clock. That's the original idea, and it comes from really sort of conceptually brilliant work from Pauling and Zuckerkandl. And unfortunately, the molecular clock hypothesis in that very pure form isn't true. Actually, organisms evolve at very different rates for all sorts of different reasons. They might be living in different habitats, they could be experiencing different selective pressures, or they might have different DNA replication machineries that just sort of make mistakes at different rates. So actually, the molecular clock ticks at a different rate in different lineages. So that makes trying to determine when things happened in the past a statistical problem, because we can have a statistical model which tries to learn about what the rates of evolution are in different lineages. And if we have a model like that, we can feed in information about these gene duplications from modern genomes and try to work out when they happened at particular times in the past. And those types of models are called relaxed molecular clock models. That's because they don't assume a single rate of evolution that's sort of been constant through time, but they do allow you to combine genetic information with what we call calibrations. So that could be fossil information that tell us about when ancestors lived in the past to estimate when these features evolved.
And what did you find?
Well, what we found was that the acquisition of the mitochondrion appears to have been a relatively late event during this process of the origin of eukaryotes. And the evidence for that, the reason that I say that, is that some of the gene duplications in the archaeal host side of the eukaryotic family tree, gene duplications associated with, for example, the development of the cytoskeleton, the endomembrane system. So that means the way that eukaryotes have many different internal compartments and can move materials between compartments, and even some aspects of the development of the eukaryotic nucleus. These gene duplications, we think, happened in some cases several 100 million years before the acquisition of the mitochondrion. So what we think is between a period of about 3 billion years ago and roughly 2 billion years ago, there was, in geological terms, a sort of a gradual complexification of the eukaryotic cell, with the mitochondrion being relatively late in that process.
So why is this event where mitochondrion become kind of acquired into the eukaryotic cell? Why is that such a big deal? It's clearly a very big deal because people are really interested in when it happened. Why is it a big deal?
Well, I think there's sort of quite diverging views on that, but for sure it's one of the key features that defines eukaryotes. One thing to say is that almost all eukaryotes today have got mitochondria. There's a really small number that don't, but it's not that they never had mitochondria, rather it's that they've lost them. And that's because they've moved into environments perhaps where they don't need mitochondria anymore. And so what the original function of the mitochondrion was, there's, I think, several very reasonable ideas. One thing to note is that around the time that we think the symbiosis between the archaeon and the bacterium, which became the mitochondrion, happened, around that time, an event occurred called the Great Oxidation Event. So that's a time when the planet went from being largely anaerobic, so with very little free oxygen for use in metabolism, to something not quite like today, but where levels of free oxygen began to accumulate, at least to an appreciable level. And that had a huge influence on ecosystems, because now organisms could use oxygen in their metabolisms and generate energy often much quicker than they were able to before. So one possibility is that by acquiring the mitochondrion, this symbiotic lineage was able to make use of that oxygen, because we think the mitochondrion would have had an aerobic, so oxygen using metabolism. The archaeal partner, perhaps not, although like a lot of details here, it's very hard to be sure about any of this. So that's one aspect. But another really important thing when it comes to the acquisition of mitochondria is that it potentially would have changed energy availability for the cell. So this is again an area where people debate. But one really important feature of the way organisms actually conserve energy or generate energy, if you like, is that it's typically done on the membrane of the cell. So organisms generate a proton gradient, so gradient of hydrogen ions over the cell membrane. And then this gradient is harnessed by protein complexes called the ATP synthase machinery, sort of like water going through a water wheel, if you like, to generate energy as the protons flow back into the cell. In prokaryotes, they've got a single cell membrane, typically the outer membrane, and that's the site at which they can harness energy from these proton gradients. But in eukaryotic cells, at least eukaryotes that have multiple mitochondrial compartments, they can actually use much more surface area of membranes to generate energy. And the shift, kind of a structural or topological shift in the way that cells could generate energy, could have been important for early eukaryotes as well. But not everybody would agree with that idea, and there's lots of different opinions there. But we certainly think that harnessing oxygen was something important that mitochondria brought to the table for eukaryotes.
So what's coming across talking to you is that not only is this one of the biggest events in evolution, and perhaps that's why it's such a controversial area. So talk me through the controversies.
It is a controversial area, and it's a very interesting area to work in. I think one of the things I like most about working on these sorts of eukaryote origins questions is that it's a very conceptually rich area. So there's a lot of different hypotheses, a lot of different ideas. What I've tried to do is come up with some computational ways of testing some of those hypotheses. But I think there's disagreements and there's debates along several different axes. Fundamentally, one of the issues is there just isn't that much data. So we're working in a relatively data-poor area of science, though in recent years, environmental genomics, where we've obtained genome sequences from lots and lots of new groups of microbes, have actually provided lots of new relevant data. But it's definitely an area where there's huge uncertainties, and so reasonable people often disagree about how things proceeded. It's also an area which has attracted interest from people with many different interests and backgrounds. So people who are interested in molecular evolution, like me, are often working on this question, but there's also paleontologists, cell biologists, many different groups of people. And often they suggest hypotheses which, in part, directly disagree, but also to a certain extent are addressing different aspects of the problem of eukaryogenesis, of the origin of eukaryotes, right? So people have different interests, and sometimes it's difficult to reach consensus. In terms of where the major debates lie, there's this distinction between mitochondria early and mitochondria late, and this space in the middle of potentially mitochondria intermediate models, which I suppose we end up supporting. And it's difficult to see, I suppose, how finally we might be able to resolve a lot of these questions. But I think one thing that's changed in the field in the, let's say, 15 years, I suppose, that I've been working on the origin of eukaryotes is that the models and the hypotheses that we have now, they make predictions about organisms which we might find in nature as we go exploring today. So, for example, in the current paper, our analyses suggest that the archaeal host cell underwent substantial complexification before the mitochondrial endosymbiosis. And nobody's found archaea that are that complex in nature. The closest relatives of eukaryotes among the archaea are very interesting, both in terms of their genomes and their cell biology. These are the organisms called the Asgard archaea. And they've got some very interesting cell features like protrusions from the cell body, which might be used to interact with other organisms in the environment. So potentially you could imagine how early interactions between the mitochondria and the Asgard archaea could have occurred there. But they're really small, so they're typical prokaryote size. And there's a huge mystery about how you go from two typically sized prokaryotes to something that's 100 times or more larger. But I suppose our scenario would suggest that perhaps if we keep looking, we will discover Asgards, which have yet more of the sort of complexity that we associate with eukaryotes. And I suppose that sort of environmental microbiology is the most promising way to find hard evidence that would really help us pin these things down.
So your work is really about the big evolutionary questions. So what gets you up in the morning? What excites you about what you're doing?
I suppose for me, this area of evolutionary work where you're using computational methods to analyze biological data, genome data. That brings together for me two things that I really like, which is on the one hand, evolutionary theory, the processes of evolution, how evolution works and evolutionary history. And then on the other hand, computers and sort of messing around with computers and programming. And those are two things that I've really enjoyed for a long time. And I guess when I was an undergraduate, that's when I first realized it would be possible to actually productively bring those together. That was at the time when bioinformatics was developing as a field. So writing some code to analyze genome data and potentially coming to biological conclusions. It's really that's always been the most exciting thing for me. And the origin of eukaryotes is such a great problem to work on because it's fundamentally important in understanding our own evolutionary history. It's a topic that many people are interested in. It's a topic with many very interesting ideas that can be tested. And it's also a topic that's often challenged us to develop new methods that better capture how genomes evolved to get better answers or more accurate answers about how eukaryotes evolved. And the hope is that as we develop better models, we learn about how genomes evolve, the processes, and we can use those to better understand evolutionary history. And I think that's very exciting for me. Yeah.
Tom, thank you so much for talking with me. This was a podcast by the Milner Centre for Evolution at the University of Bath. I'm Turi King, and thank you for listening. If you have any thoughts or comments on this or any other episodes, please contact us via our social media. For more information about the Milner Centre for Evolution, you can visit our website.